Welcome you all to the opening of phytoteratology, which is a project that I've been working on for the past 2 years. It's a labor of love towards the plants, and it's an expression of this desire to know the other. So I've been exploring, as I was talking about a bit yesterday, I've been exploring the cryptic life of plants for a while now, even though science is opening them up and their inner processes, and also uncovering their mysterious complexity, which always comes off as a bit of a surprise because we consider them so simple and so purely exterior and like an open book. And so this is the third artwork that I've done in collaboration with plants. The first one was Primaga, which explored the trans-species of plants and humans. It was more speculative. What if the human species was not just fully human, but had a complex life cycle? So as a part of the life cycle, you would actually exist as an algae. And it has a lot of implications, but one of the notions was a bit of a critique of transhumanism, of this idea of the almighty mind and this aspiration towards ever more abstract and complex forms of existence, which we as humans feel somehow endowed to having, as opposed to other more primitive life forms. And plants are actually, in our Western hierarchical view, still considered very much primitive, and that's where the surprise comes from. They communicate, you know, they can hear. Oh wow! And on one hand, it's coupled with this fascination. On the other hand, I think in this fascination there's a lot of abuse that's going on, because we are looking for parallels, and we're looking to find something that is like us in order for us to legitimately raise them up to our level. So having all these complexities in mind, I started working on this opus, which I titled Confronting Vegetal Otherness, which I would now like to actually change the name. The confrontation was something that I felt at first compelled to use because of this ultimate foreignness. I've been proven by Michael Marder that philosophically there is no legitimate way that we as humans can have an empathic relationship to the plants. All the empathy we feel towards them is a result of the process of anthropomorphization, in the way of finding traits of humanity in the plants that are not really there. So the challenge was then what kind of relationship do you establish to someone where it's not fair to them to look at them as if you were like them. I turned to the materiality of this possible interaction and to this emerging field of science and humanities called biosemiotics, which looks at physical properties and as molecules, as carriers of meaning. The meaning is only formed in the context of the organism. And I asked simply, what can I as a human, in my time scale, in my ways of being, in my animality, what can I produce physically or mentally even? It doesn't really matter, but that the plant could sense me, could integrate the information of my presence into whatever it made. So the first exploration was culminated in this performance. It's a durational piece simply because that's the time scale that the plant's needs. So I was standing for 19 hours and throwing the shadow onto plants, onto germinating press.
You were standing in front of the plant or something and drawing a shadow on it?
Yeah.
For
Very
how much?
simple. 19 hours.
You were standing.
Yeah. I took breaks. It was not so much about human endurance, and it was not about, you know, even per se having a sort of a spiritual
You just stand.
encounter, but it was more about the discipline of submitting and committing to this intercognition, what it takes for the plants to recognize my presence. In fact, I did sort of, after doing a lot of research, which was enlightening, but then coming to the conclusion, okay, so we know all these different things that plants, as others, are telling us. How do I, as an artist, react to that? How do I make something out of that? And it's very, very constraining, very restricting. But I did this analysis, which is serving sort of as a guide of exploration, that one of the differences that I chose to focus on in the plant-human relationship, or difference, or similarities, was the openness of the system. So, and by how closed it is, I mean how circulatory, how self-contained and it is, how important it is that we have the heart, brain, the limbs, and how this is all connected in a system that cannot simply be divided. And at the level of cells, the unit of the cell is actually kind of similar both in plants and animals and unicellular organisms. It's very difficult to just randomly split the cell apart and have the two cells survive because of mitosis, etc. But many similarities we have on the level of cells with plants. And then as we grow, raise the level of complexity, we come to this organism, which we are so concerned about, because especially in our culture, it's all about the individual and then sort of the structures which eventually subjugate this individual, and the struggle is real. But our focus, and you know, especially as artists, is from this. And on the plant side, there is no individual as such. It's a different type of construction, and it's been philosophically more or less bothering everyone that has thought about plants, from Aristotle to nowadays. And nowadays, they're sort of rediscovering the plant as an embodiment, like a living embodiment of the anti-metaphysics, because they just do not fit in with the system that a thought that we have constructed. So the level of the individual presents sort of a conflicting relationship, one that is a bit absurd, has its own humor, and presents the biggest obstacle in understanding plants. That's my hypothesis. So both of these pieces, Phytoteratology and Scotopoiesis, was an enactment of this clash, the question of individuality. In January, from the 6th to the 19th, I'm going to address the level of cells. I'm going to pursue with a couple of colleagues in this open lab / exhibition setting the co-culture of plant and human cells. And when we look at human cells, there's an interesting equality or loss of this immediate hierarchy of more or less complex between human and plant cells. They're more or less units whose soul is not as graspable. They're divided, they're small, they're morphing, they can be changed. Stem cells can be forced to grow into an organ or even another organism. There's this plasticity that is retained in cells. Of course, it's all artificial.
Also for plants, same thing with plants or human.
Yeah, And that's, I think, an interesting situation, because we have actually a lot of problems with understanding what a human is when it's only in a petri dish. And so finally, as a relief, so that's that exploration. But in this one, I wanted to become a bit more emotional, empathical, with exploring the concept of what it would mean to mother a plant and what it would take to perhaps have the plants contain a bit of an imprint of me as well. And I know that, so for both scotopoiesis and for phytoteratology, I'm kind of playing the human. I'm not resisting what comes naturally or culturally to me. It comes culturally to me as the human, which is this longing for finding a place in the other, even in this vegetal other that is devoid of an individuality, and yet precisely because of this can continue. It's in a way immortal, and in a way it accepts because of this process of assimilation of the sun, of the nutrients, and of this endless growth. It has an endlessness to it through also all the organisms that are surviving based on it.
It's always been like that, and it's always going to be like that.
But in plants, we sort of recognize that in ourselves much less so. So when we exist in a system of a strange paradox of hyper-individuality and complete diminishment of sort of as a almost careless to what our needs are to feed the system in this like post-Fordist capitalism, there is maybe some peace that we can find in plants and their existence. So it's looking for a primal material connection with plants. So the project, maybe you can follow me and I will show you. So one of the key findings when doing research was that, of course, we've all heard of phytohormones and how they affect human beings, and that's one direction. The other one is also present. In fact, human hormones can affect plants. Moreover, plants have hormones that are identical to human hormones. And they are, this was basically overlooked in most of the sciences up till very recently, because it was preposterous to think that, plants and animals would share the same hormones. They're very different. So it's a bit of crossing of this divide that science is doing and saying, okay, look here, they have also steroid hormones, maybe not in these huge amounts, but the metabolism, the metabolic overlap is still there. So they've proven that steroid hormones affect the morphology of the embryos. And the first step, I guess, was to try to isolate the hormones from my body. And it seemed that urine is an uninvasive way. It uses these columns, and this is kind of a milking machine that I did that I also use in workshops, because you administer the urine here and you actually have to pull down on this. It creates a vacuum and it sucks the urine through, and the steroids are left on this column, and then you take the column out and you put it in this special dryer. So what you're left with is this essence, and this essence has a very strong smell because it contains what you would call pheromones, actually. It's really interesting then to have people smell each other's extract because they react very, very individually to smells. Some, what is pleasing to some may be super repulsive to the others. And I've worked also with embryos of sea urchins, morski ješki, as a model to see the effects of the steroid extract. And in fact, if you administer the steroid extract to them, they change morphology, they become more round, and so it's affecting their development, the early development. Sea urchins are animals, so they're much more sensitive to this than plants would be. But I also appreciate the resistance of plants in this sense. This was the first part, and I had it analyzed. I found that it's mainly steroid metabolites that are present, and they're detectable, but only for species of different steroids I was actually able to detect here. The experiment is very unscientific because the urine, every time I pee, there's a different ratio of things. There's also other substances mixed in. But it's this complex snapshot of my excrement? Is it excrement?
Let's say, yeah, okay, maybe.
Excretion that is then forming this link. So it's similar to what would happen if you went to pee on the grass. But in this case, it's really framed quite sharply and quite purely through this steroidic molecules as communication. The process on the plant side starts with Arabidopsis. Arabidopsis is the small weed which you probably didn't notice, but it's right out there. It's a tail cress. Even though it's ubiquitous, many languages don't even have a common name for it because it's just this weed. And this is quite ironic because it's so popular in the sciences. This is the most transgressive plant on the planet, maybe one of the most progressive beings. It has had everything done to it, every possible thing with the mutations, with its genome was sequenced, and it's used all over. It's a bit of a slut in this sense because it will produce, it will flower, it will produce seeds quite reliably, and that's why it's the small organism used in science and very rarely anywhere else. And this is the plant that I used because I was also a bit opportunistic. I went to the Sylvius laboratory in Leiden where they're actually researching how to make this plant, which is 1 year, so it's an annual, not a perennial, but it's an annual plant. So they're trying to make it have several cycles of flowering and growth. So they know how to do this so-called somatic embryogenesis, which is basically a process of starting with a bunch of cells and then from a single cell, one of those, creating this new embryo. We would call it cloning, and when you do it in plants, it's called somatic embryogenesis. And the starting point is an abortion. So you take a fruit, which is, it's called a silique, of this Arabidopsis, and you take these embryos, which are just 11 and precisely 11 days old, and under the microscope.
Like today, 11th of 11th.
Yeah. So it's on the 11th day, you take them out of their seeds, and then you transfer them to this medium that has hormones, but plant hormones, actually a pesticide. And this pesticide causes the embryos, which are immature, to develop these tumors or callus. But this callus is special in the sense that it is just dying to become embryos again. And after 2 weeks of this treatment, I transfer them to a medium that contains my hormones. And so out of this mass, several embryos will form, and these embryos are then also shaped by the presence of the hormones. So the absence of its plant-like hormones and the presence of more mammalian, but let's call them steroid hormones and metabolites. What is the result? It's these plants. These are the magnified images of some of them. I have a really beautiful collection. But I'm showing them also here live. These are incubators having these embryos at different stages. So these are from Tuesday. These are embryos.
And they're growing.
They're from Tuesday. You can see them. They're the smallest green torpedoes. They're in the torpedo phase. So you can kind of see the two leaves and the root, what will become a root.
And they're just growing by themselves here without anything else.
This is a medium that contains vitamins, minerals, and steroid hormones. And they have the light. The temperature is kept to 20 degrees. It's in a sterile environment. They have also a bit of sugar because as soon as you have them in a tissue culture, they always need a bit of sugar. Plants, as you know, usually produce all their carbohydrates with the use of the sun or the light. And then you have them at different stages. So sometimes
These are the smallest.
these are the tiniest. Sometimes they only form this callus, this tumor-like
And
state.
how old are they from this? How much
These are about 2 weeks. And you can see some of them did not develop, so they remained in this tumor
Some of them
state.
develop, some they don't. That's usual, isn't it?
Yeah, and also to get this, it's extremely labor intensive to get the embryos because it's kind of amazing that we as humans are even able to do that because it's such a fragile organism at that point, and you're handling it with a sharp needle and some tweezers, and you have to gently squeeze. And I'm still surprised.
Why
that
do you
it
have to
happens.
squeeze them?
The way that over there where
I know, I saw that.
And you have to collect around 400 for one of these experiments, and that's about 12 hours of work. So it's actually quite intensive.
But after that is also a lot of work after you get them here, or not so much?
This is a system that is self-sustaining. So the light goes on and off, and there's cooling that goes through here, and is it called entrails? The internal, the life support system is in the back, and it consists of refrigerators, ice makers, and this electronics. So once it's here, the only thing that we have to fear is infections, which you can also observe over there, the two beautiful fungal colonies that are growing, which will quickly overgrow the whole.
That you cannot somehow protect them from fungus and things. Sometimes you can't.
No, I mean there is always an option of using antibiotics and fungicides, but we know that no. Anyway, it would affect it affects in another way the plants. But basically, the only way to get rid of them is to take everything down, and you have to kill the plants if you don't want the fungal infection.
When you look at them already here, you can see it's a little bit like a plant. But did you notice it's something? Does it look a little bit like you?
Yeah, I think so. It has definitely a lot of green, like I have green eyes. No.
That's true. I mean, plants will have green. That's obvious, isn't it?
No, of
No?
course not. But then there's definitely another type of nurturing feeling that I have. I've effectively
Yes.
separated them from all the other plants. Now they're mine.
Yeah.
And that's the only real thing that I can say about how they are different.
Okay.
They have an
Understand.
amazing variability. After this procedure, each one looks like, and you can recognize them because I've been shooting them following them several days, and you totally know that, this is this one. Oh, yeah. Oh, wow, you're changing. But this is due to the fact that in the beginning, they look quite different. I could say, yes, of course, they're unique, but so is.
But
but
do you
so are all
think
the
that
others.
if you didn't put your hormones in them, they would also be all different, isn't it?
Yes.
If you just let them grow by themselves, it will be the same, no?
Yeah.
So
But
that's
it's not the same. And
Probably
even though
it's not. That's why I'm asking you. Where
I-
is that? How
I-
do you notice that difference?
I know the difference.
Just maybe, yeah. You just know it. Yeah.
I just
okay.
know the difference. And that's ultimately, I've been asked this by the scientists, have you observed anything? And I'm looking at them, do you know your material? You know how huge the natural variation is. And the scientists never notice it on one. They do statistics. And then they look for significant difference. And no, I did not do that. So I know that the environment, especially molecules that have their own receptors, and it's been proven that they change this, the epigenetic pattern, and the plants are forever influenced by this. But they're also influenced by many, many other things, more significantly probably. And yet, this link persists. So what happens if I take these plants and I have them with me for years to come and continuously do this? Or if I take a plant that does not have this one-year cycle, that is more like a bonsai, and I continuously do this and I travel with it. And, so that's violence, the plant principle. But there is a gap that I filled with my desire and my material. And that's all I can say.
I understand. That's artistic project after all, isn't it? Art project, no? Not scientific.
Well,
Yeah. That's it.
do you have any questions?